2023. I'm way ahead, but that's why we're here, to and move forward. Talking about the future, we have a short video about the past, about yesterday and what has happened yesterday, and afterwards Amy will introduce you and will introduce her and the session ahead. So fun and see you later.
Welcome to Tromsø, the Arctic capital.
Right, hello everyone. Welcome back to the third session on plastic pollution here at Arctic Frontiers. My name's Amy Lusher. I'm a senior research scientist at the Norwegian Institute for Water Research, and I've got the honour of chairing the session today. We've got a variety of speakers, but I'm going to take 5-ish minutes and just put some perspectives in place before we start. I hope you don't mind. So I think over the last few days, and actually over the last few years, it's been really interesting to me to see how the need and the requirement for certain methods has been developing. And a lot of our presenters yesterday and even at the poster session were talking about how important it is for us to have the right methods, methods that work, and also look for what we can work with now and what we already have that's available, etc. So I wanted to reflect on where we were with the Arctic research. So a little bit of background, which I forgot to put on my slide. I've been lucky enough to work in the Arctic since 2012 on microplastic pollution. And it's been incredible to watch the development of our research over the last few years. But it's still very important that we're using methods that are tailored for the Arctic environment. Sometimes we can't use the methods that we've developed in other areas of the world. We have things like very challenging shore conditions, often lots of different anthropogenic activities that we need to take into consideration, and then we also have lots of fauna that we will need to work with in a different way. So most of the methods that have been developed have been south of the Arctic, and we need to optimize what we're doing up here. I think it's important that we're using an approach which is harmonized so that we can all use similarly related methods, but we know how different they are and how they compare to one another so that we can generate data that is comparable and that we can use it to build a really big picture of the whole scale of the Arctic and what plastic pollution means to us. So at the moment, or historically, we've mostly seen differences between different research teams, different countries and regions. And this is causing consequences for monitoring, risk assessment, and legislation. Because some of us will argue we don't have enough methods, or we don't have the right methods. But I don't think that's true. We do have methods. We just need to know how different they are and which bits need a little bit of changing to allow us to be comparable. So this is a statement I use in quite a lot of my presentations, and I think it's super important because in order for us to reach substantial improvements in environmental sustainability and socio-economic development, it's essential for us to undertake major actions for the evaluation and the optimization during our microplastic or plastic litter assessments. One of the most important steps forwards that I've seen in recent years has been the work that's been undertaken through the Arctic Monitoring and Assessment Programme. And through this, we've been seeing the development of recommendations for monitoring programs and lots of work linking different efforts across the Arctic to understand plastic and microplastic pollution from the local scale, but also for the breadth of the Arctic. You may have seen this slide previously yesterday. I know Katrin had it on her slides. And these are the recommendations that have come forward from the AMAP working groups. the prioritization of different environmental compartments for immediate trend monitoring, baseline mapping, future trend monitoring, and then for comparative assessments for later on in our work. We still have a lot of work with the methods, for example, with marine mammals. I think it's necessary to also highlight that we were asked to try and choose a few matrices within the AMAP group, and we couldn't choose just 3. That's why we put this prioritization together, because it's really important to generate our data across different matrices rather than just focusing on one. So out of one of our recommendations, and you may have seen this on our posters yesterday, is that we recommended a joint sediment and water approach because we knew that it could be carried out through the same sampling campaign, and we could provide complementary but not overlapping data, and we could provide the most complete picture of plastic pollution with relatively stable methods. For example, we can use the water to track potential rapid changes and fluctuations and influx of plastics. And through the sediment, we can look at spatial or temporary integrated signals. However, I must note that quite a lot of our work is still biased on the marine environment. We are needing to get ourselves together with our freshwater and terrestrial samples as well. So I'll just wrap up with this last slide and just say that there is a way that we can address our different methods by breaking it into different pathways, so from sampling all the way through to data reporting. And if we can identify what's comparison between our methods this way, it allows us to optimize and improve at different elements. I think Janneke might talk a little bit about some TRLs maybe in her presentation later, but I'll pop back to that. So moving forward, recommendations for monitoring strategies. We need to consider the policy question that's being addressed, as well as any of our resources that are available. So we need to ensure that we're looking for cost effectiveness, so we can maintain monitoring programs. We want to be able to prioritize and address significant risks and associated indicators, encourage cooperation, favor innovative and opportunistic approaches, and build on existing monitoring approach activities. So I'll end by saying that I believe harmonization is the flexibility to adapt to scientific, logistical, environmental, and ethical constraints, and it definitely takes everybody in this room and everyone who's not been able to attend with us to address the issue of plastic pollution. So with that, I'm going to say thank you for everyone for coming, and I'm going to invite my colleague Anthisa to the stage so she can talk to you about her work on modeling microplastics in the Arctic.
Does it work? Great. Thank you very much, Amy, for introduction. So I'm going to talk about modeling approach in microplastics assessment in a natural environment. And my presentation is about modeling the influence of biogeochemical processes on microplastics transport in the Arctic Ocean. So we all know that microplastics are transported in the ocean by ocean currents and by ocean mixing and turbulence. But in this work, we wanted to focus on how biogeochemical processes affect microplastics transport in water column through biofouling, ingestion by zooplankton, and sedimentation in detritus. So for this, we developed a numerical model that simulates concentrations of microplastics in different forms, considering biogeochemical conditions in seawater. This is just an overview of model setup that we used for this work, and this is just to show that we combined plastic model and combined it with Oxidep biogeochemical model through special interface called FAB with transport models. We used the two-dimensional model for Oslofjord and then applied the model setup for Arctic Ocean using ROMS-20 three-dimensional model. So we called our plastic model Bioplast. It considers 4 groups of microplastics. This is particles in virgin form, MP3, particles fooled by phytoplankton, MP biof, particles consumed by zooplankton, MP head, and particles in detritus, MP dead, And bioplast receives information about background biogeochemical conditions from biogeochemical model Oxidep. First, our step was to apply this model setup for Oslofjord, where we have reliable data on the source and background concentrations of microplastics. This red circle shows the position of the source in the part of Oslofjord called Bekelag. This is just a wastewater treatment plant. We applied a two-dimensional model for this region. It simulates vertical and horizontal transport of organic matter and microplastics on the small scales, and it considers water column and sediments as one common computational domain. So here are the main results of our model application. This is background variability of biogeochemical parameters in the model. So the main features is phytoplankton bloom in summer period, followed by zooplankton development and the flow of detritus through the whole summer period. Upper panels show the water column, and lower panels show the sediments and bottom layer. And here are the results for microplastics. We see seasonal variability in concentrations of microplastics with natural buoyancy, so it means that microplastics in this case are considered as a passive tracer. We see the purification of the surface layer, and particles, when they are biofueled, it increases their vertical velocity quickly, and they start to sink to the bottom and accumulate there. Here's the case for floating microplastics. They concentrate in the surface layer, but in summer period they are again transported to the bottom due to biogeochemical processes. And negatively buoyant particles that initially sink, they just really quickly sink to the bottom, and they are not affected by biota in this case. Then we applied this model setup for the whole Arctic Ocean. We used ROMS 20 three-dimensional model and considered two scenarios. Microplastics were supplied from Arctic rivers, but just only Russian Arctic rivers. And in the second scenario, microplastics were supplied from the North Atlantic. It is the western part of computational domain. And here are the results for application of this model. This is spread of microplastics from Russian Arctic rivers. You can see that they spread really fast across the whole Arctic Ocean, and we see this green hatching corresponds to areas where there is phytoplankton blooms, and red hatching represents areas of zooplankton development, and we see the corresponding change of concentrations in different forms of microplastics. And here are the results of spread of microplastics from the North Atlantic. We see that microplastics go along Norway Zemlya archipelago and through Kara Gate. They spread to the Siberian Arctic. Here are the dynamics of microplastics concentrations in near the Svalbard archipelago in Greenland Sea, and we see similar picture as it was in Oslofjord. So biota contributes to seasonal changes of concentrations of microplastics and to temporal purification of the surface layer, but phytoplankton blooms are too short to affect microplastics buoyancy, and there is no significant transport to the bottom here. This is results of on the 8 year of simulation. So this black hatching is the area where Atlantic plastics prevails, and gray hatching corresponds to area where river plastics prevails. And we see that maximal concentrations are absorbed in the area of Atlantic microplastics, and it fits well with our results of our observations together with Svetlana Pakhomova and Igor Zhdanov, that maximal concentrations are observed in the Barents Sea. And I would like to talk a little about assumptions of our model that cause some limitations in the results. So we consider only fibers of a certain size, and the other properties were not taken into account. We do not consider now biodegradation, but we plan to include it in our model. Also, we set constant vertical velocities for the main variables, but we also will improve it in the future. And the main assumption is that only phytoplankton in our model causes biofouling, but it depends on the biogeochemical host here. And a little about our future plans. We want to add sources from Pacific Ocean and maritime activity in Barents Sea. And the most important, we want to parameterize microplastics transport with ice. So thank you for your attention. We are looking for collaboration, so please don't hesitate to contact us. Thank you very much.
You can stay up here while we ask some questions. Okay, that was a fantastic presentation. Thanks, Anfisa. Do we have any questions from the audience? Have a look around. No? Are there any in the online forum at this point? Probably not. Whilst I'm here, whilst people are thinking about questions, so we have a short amount of time, you can use the online forum for those that are listening in to us to add additional questions, and we're keeping an eye on that in the chat. I have a question for you, as we have no questions here. Regarding your assumptions with the fibres, how do you think your model might change if we were to replace the fibres with a different particle type?
Yeah, thank you for your interesting question, Amy. So we will change parameterizations of biofouling because we adapted our parameterizations for fibers, and it has some certain rate of biofouling now, but it is really difficult even now with fibers because we don't know exact biofouling rates, and we don't know exact rates of ingestion by zooplankton. So there is a high degree of uncertainty here. Yeah, so we need data from biologists and some experimental data on this biogeochemical processes there. Yeah, so we are looking for collaborations in this field.
Shameless plug. Fantastic. Thanks for your present question. And if there are any other questions after the presentation, we've got to move on to the next speaker now because we've got a very tight schedule. But I'd ask if you can ask Anfisa in the lunch break and find her. That would be fantastic. So thanks, Anfisa.
Thank you very much.
Okay, so our next speaker is Anna Mir from Norse. I'll invite you up to the stage. She's coming from this side for me. And she's going to talk about some of the work she's been doing with some novel methodology for detection of micro and nanoplastics. Thank you.
Yes, thank you. I am going to present a project that is a collaboration between the University of Tromsø and Norse. This is a part of several projects we are having where we are combining advanced microscopic methods and novel gene technology methods and studying some impacts or studying some pollution and connecting this with responsible research and innovation. In this project, it's on detection of nano and microplastics in fish. And I'm not sure how I'm going to turn to the next slide. This one? Yeah? Okay, sorry. So, I guess today and yesterday you have been talking about plastic waste. As we know, there's a lot of plastic waste in the ocean, and we can see some of it. And in this project, we are more concerned about what we cannot see, which are at the microplastic and nanoplastic level. We know that the ocean degrades the plastic. It's the waves, it's sunlight, it's maybe sediments that degrade the plastic. And we also know that fish can consume plastic by eating, for example, smaller organisms, or get it through the skin by just being in the water where there are micro and nanoplastic. It's an assumption that this have a negative physiological effect, especially the smallest particles, but there's still uncertainties around this. This project was started based and inspired by some studies that was done a long time ago, where they were introducing plastic beads to fish cells. And then it was with skin epithelial cells, but then not in the fish, but in fish cell culture. And what was found then was that the particles were taken up and covered by cell membrane. This was not studied so much at that time. The microscopic methods and so on was not that good as it is today. So we started up the new project with much more advanced methods. And what we do here is that we do not collect fish in the oceans, but we use fish cell culture and we use salmon as model species. We make different plastic by ourselves at the nano and micro level, just to know the size of it and also the origin of the plastic, because plastic can be made of many different things. What we do, what we have done so far, this is a slide of the experimental flow. The University of Tromsø and Nofima has a very good, excellent facilities in Kårvika, where you have different types of fish. But we do the sampling there. And then we make the cell culture by ourselves. And in this project, we focus mainly on skin cells. And then we add and expose to different micro and nano-sized particles of plastic. We have also used gold just to see. And we also have labeled this to make them visible by using microscopy. So this is how far we have come with the research we are doing in the lab at the moment. I'm going to show you some of the pictures of what we have found. It's just fascinating what they can do by this advanced nanoscopy. And here you can see that we have below is a 3D figure, and on that side we have a 2D figure. And what you can see, what is colored in turquoise, is the plastic particles. So, and then you can see what we thought. Then you can see the cell. You can see the cells here, around here. And then we thought that in the cells that we thought that when they were taking up micro and nanoplastic, that the lysosome would have degraded this. But as you can see, they are colored pink, and they do not degrade the plastic. We can still find it in the fish cells. So, and the lysosome is interesting because they are kind of the garbage central in the cells, and the kind of down treating, for example, viruses, bacteria, and downgrading this. So we can find it in the cell. So what we will do next now is to use more kind of different omics methods to study if actually the presence of these plastic particles in the cells affect, for example, gene regulation or other things in the cell. We will also analyze the result related to the plastic type, because we have a hypothesis that different origin of plastic might have different effects. And we're also discussing the broader issues. By working, for example, with nano and microplastic, we're making them by ourselves. So we are discussing HES issues related in the lab for those scientists involved there. And we also discuss, have a focus on RRI. And RRI, very short introduction to this. It's a method. It's a kind of you are involved discussing ethical, social issues as long as the project is going on. You are working together. You are reflecting on the project's approach, engaging, including stakeholders, and so on. And a purpose in this project is to assess the potential implications and societal expectations with regard to this research. So here we have had some internally workshops, both to strengthen cross-disciplinarity, because actually it's 3 different disciplines in this project. You have, or actually it's 4. You have a fish immunologist, you have molecular biologist, and you have those working in microscopic and nanoscopic, and more social science who are working in this project. We have had some workshops where we have used ethics cards as a part of a tool of the RRI to discuss different aspects by the research we are doing. We are now planning to have a workshop with other researchers connected in NORS. NORS have a North Atlantic Plastic Center where we have a lot of different scientists working together with the plastic, and also an engagement internationally to have this kind of broader, have other researchers working with plastic. And finally, we will also engage more broadly outside the project. And we do not have a homepage, but we are on ResearchGate, and the project is funded by the Research Council, and it's a collaboration between the university in Tromsø and NORS. Thank you.
Let's stand up here. Thank you. That was a lovely interaction. I've never heard of that before, so I was writing some notes. Have we got any questions in the audience? We'll start at the front then here.
It's not easy to see the people in the
really
audience.
Hard.
Hello, I'm a colleague of Amy in NIVA, and I'm curious. I didn't understand what kind of microscope did you use, and what is the technique?
They've switched the slides over now, so.
Oh yeah. Oh, sorry. Yeah. It's a very advanced microscopes they have made partially developed at the Institute of Natural Science and Technology at the university. So, and you can take picture both in 3D and 2D, and with different colorations, different backgrounds. You can really play with the model depending on what you want to see. So,
How do you put the color? I mean, how do you?
That's how you work with microscope. You have different background colors in the sample you see, just to have the different contrasts. So, and of course that's a tool in itself, and expertise in itself by working with this, and a lot of adaptation that is going on. So I can make you in contact with the people that is really doing this. Yeah.
Trying to see if there are any more people. If you do put your hand up, can you wave it like a lot? Okay, I can't see any more questions. So what we'll do is we'll move on to the next speaker. Thank you so much for the talk. And if there are further questions, please do so in the lunch break.
Yeah.
Thank you, Anna. So I can invite another one of my colleagues to the stage. Dr. Jannicke Fogh Andersen is our colleague who works up here in NIVA Tromsø, and I'm going to let her talk about some of the work we were doing this year on macroplastics.
I'm going to talk about the big stuff. We were asked by the Norwegian Environment Agency to give some advice on how they can develop a monitoring program for microplastic in litter. And the background was they saw a need for monitoring of litter and microplastic to secure that the measures that they implement are knowledge-based and that they can detect changes over time. And the report was out just before Christmas, so it's available on their webpage. So what we did, we identified these international obligations and national needs. We looked at the methods available and their technological readiness level, and we rated them according to whether they are at the basic research stage or at any stage towards the process of being able to be implemented. Then we looked at monitoring data available in Norway, and we tried to see if they meet these obligations and needs. Two things I'm not going to talk about today that we did was that we also identified activities that could provide additional data, and we estimated relative costs of different monitoring alternatives. And based on this, we gave advice on a monitoring program. So the size fraction we're talking about is items larger than 25 millimeters. We also included items that are included in beach litter protocols that are a little bit smaller, such as cigarette butts. And we only looked at the natural environment, coast, ocean, lakes, rivers, and we were also asked to say something about the northern fulmar, as this is an established bioindicator, and they normally eat the meso plastics, which is between 1 and 25 millimeters of size. We did a literature study and arranged a stakeholder workshop with Norwegian stakeholders to identify what are these international obligations and national needs. And we identified a number of litter classification needs to identify specific items. There were a reference to some specific protocols. The OSPAR protocols and the EU joint list is the most relevant for our region. Also, the need to identify these single-use plastic items that are now under regulations. Items specific to the Arctics, and in order to implement extended producer responsibility, we need to be able to separate items from commercial fisheries and recreational fisheries and aquaculture, as well as identify polluters and producers. And stakeholders were also, they also wanted to know something about litter items of high concern. In the marine environment, there are lots of requirements set for measuring the amount of litter, normally in number of items, and also be able to follow the changes in composition and source of this litter. So this goes for beaches, floating on the ocean surface, water column, seafloor, and biota. And then we are seeing in the pipeline that there are becoming more requirements for also monitoring litter in non-marine environments, both the amounts and the sources. When we assessed the monitoring methods available, one of the key limitations is survey design. We rated this as having a very low technological readiness level. How do you select representative sites for monitoring, especially in Norway, where you have such complex morphology, both above and underwater? When it comes to litter classification systems, we have protocols that are meeting many of the management's needs identified, but there are some further development needed to meet all needs. We also saw the methods are well developed for marine compartments and the northern fulmar, but we see development needs in biota, freshwater, and terrestrial systems, and we need a harmonization of methods across the different environmental compartments. So this is the monitoring data we have in Norway. We have 7 OSPAR beaches. We are doing sampling of the northern fulmar stomachs. In the Barents Sea, we're doing trawl surveys, and they are applying their own protocol, but they are going to be harmonized towards the one recommended by ICES, and they're also doing surface water observations. We have the International Bottom Trawl Survey in the North Sea that we take part in, and there are citizen science data on beaches using a protocol adapted from the Ocean Conservancy Protocol, which is internationally established. When we looked at other data available in Norway, they are mostly marine, they are single short-term study, they use a variety of protocols, data is not accessible, and we see some citizen science initiatives in non-marine environments, but they also have the same kind of problems. So does the monitoring that we're doing meet these needs and obligations? We are applying the OSPAR beach litter protocol, and we are applying the citizen science protocol, and that meets some of the specific requirements related to applying protocols that are established and harmonized, and also the identifying these single-use items. We are not applying protocols that can identify items specific to the Arctic. And also, we are not collecting data that can be used to implement extended producer responsibility. And we have no idea what even these litter items of high concern are. When it comes to measuring the amount of litter in the environment and looking at the composition and sources of this, we do have some things going on. We have the OSPAR beaches, we have the citizen science going on, we have the Barents Sea visual survey on the surface water, we have the Barents Sea trawl survey and this North Sea trawl survey, but the coverage is not good enough to be representative. And then the Northern Fulmars, again, not representative, and we're not documenting any other impact on biota. And for the non-marine compartments, we're just not doing anything. So, advice on the Norwegian monitoring program. We suggest that you should apply the EU's joint list of litter items where possible. We also encourage Norway to contribute to and develop harmonized protocols across compartments. And also develop protocols to identify these different items. We can separate out commercial versus recreational fishing and aquaculture, being able to identify are there any items that are specific to Norway or the Arctic that we should monitor. And also if you're going to have any knowledge about who's going to pay for cleanups, as is the case for implementing extended producer responsibility, you need to know something about the brand name, age and geographical origin of litter items. And then, big take-home message, you need research to identify what kind of sampling design should you have on a monitoring program. And on the good side, we can meet many of the current management needs through beach litter data and increasing the number of OSPAR beaches, including increasing the frequency of sampling. Most of them are only sampled once a year. The requirement is four times a year. Depending on the snow and ice conditions, maybe we can get up to at least 3 times a year. Also, we strongly encourage that the experts are involved in citizen science initiatives to secure data quality and improve the relevance of the data collected. And we also recommend to develop and extend monitoring to all environmental compartments. So, on that note, and with these beautiful pictures, are there any questions? Or do you want to ask that question?
You can ask the question if you want to. That was an excellent presentation. Thank you.
Thank you. That was like a 111-page report in 8 minutes.
8 minutes on time, exactly. So that does mean we do have time for questions. So if there are any questions, can you wave frantically? I've spot one right there.
Thank you for the presentation. What's exactly an OSPAR beach? So what characteristic needs a beach to fulfill to become an OSPAR beach?
Yeah, so there are some requirements related to picking OSPAR beaches, and they should be like sand or pebble beaches, and they should be 100 meters at least. We might have some challenges finding those kind of beaches in some areas of the Arctic. And that's also one of the AMEP recommendations, actually, is to be a little bit more flexible in being in the choice of beaches so that you can adapt the method to Arctic conditions.
Okay, thank you.
See, we have time for one more question if there is one. I'm going to take silence, that's okay. Thank you again for your presentation. And any more questions later on will be great. Thank you. So next I'm going to ask Marta Haar to come to the stage. She's from SALT, and she's going to be presenting on some of the work she's been doing that leads on from Jannike's presentation about the choice of sampling and survey designs.
Thank you. Okay. So I'm just going to jump right in for the sake of time, but I'd like to share some contemplations from and results from 2 data sets that I worked with recently. The first one is a fairly large data set from Norway and the Barents Sea region consisting of 288 beaches sampled in a nested stratified random design. And the second is a review of 149 papers reporting marine litter densities around the world. And starting with the beach litter data set that I collected myself, working with marine litter means working with a lot of variability in space, in time, over different scales. And these graphs just show examples of spatial variation on a regional scale. The donuts here show the prevalence of clean and polluted beaches in different regions sampled around the Barents Sea. And the map here shows the proportion of beach litter originating from maritime sources in different counties in Norway. So we have regional variability to consider during survey design in terms of both replication and things like, depending on the scope of the study, you could end up with spatial variability being a significant source of noise, a confounding variable, really impact the external validity of your study, so sort of defining your regions is quite important. But it's not just over sort of the larger regional scales that variability is high. Small scale variability can also be really high. So within the regions shown on the previous slide, sampling was done in a nested stratified sort of random manner here. And while we did see detectable differences between, like among regions, like I showed on the previous slide, the spatial structure of the data actually explains a fairly small portion of the variance. So you can see on this variance components analysis, it's only about half the variability here that can be attributed to the spatial scale, or the spatial structure. So we have all this variability that is just really high local stochastic variability, and the actual regional differences that you saw on the previous slide is a very small portion of the total variance. And obvious spatial variability at different scales just then, of course, begs the question of what that underlying spatial structure is. And this is often investigated through, for example, a semivariogram analysis, which in a nutshell is a graph showing dissimilarity among pairs of location based on the distance between them. So what you expect to see is that sites that are really close together are not that different, while dissimilarity increases as the distance between them increases. And eventually this curve will flatten out as distance ceases to predict how different or similar or different sites are. And the point, this inflection point, you typically call the range, which generally then reflects the scale of spatial autocorrelation. So far, though, I haven't not managed to do anything meaningful like this from marine litter. This is my semivariogram from beach litter in Norway. So you can see there's no pattern whatsoever. Complete lack of pattern at a larger scale. And you see on this small range, which this plot is just zoomed in on that, actually see at times greater variance dissimilarity among sites that are really close together than those that are further apart. And another attempt at describing spatial patterns in this way in the density of floating litter in the Pacific also failed to fit a meaningful variogram model. So there could be lots of reasons why we haven't been able to describe this, but what is clear is that there's a lot here that we don't understand about the distribution of litter, and that we kind of should try and, or need to keep working on figuring this out. As the scale of spatial autocorrelation, it's quite important in terms of determining like sampling units, the size of these, and avoiding pseudo-replication and those sorts of issues. For example, when you have a spatially heterogeneous distribution, if your plots or sampling units are smaller than that scale of patchiness, it's going to be prone to both spatial autocorrelation and high variability. Of course, if your plots are larger than the patch size and the distance between them, your units you will be less variable, but you're not going to be able to detect the patchiness. So you kind of need to, what is important for your research goal will sort of vary. But also importantly for marine litter, what happens when the plot size itself is variable? In ecology, we generally avoid this, and sampling methods will be either area-based or distance-based, the latter meaning that you record the area distance it takes to reach a certain density or count. But if neither is standardized and plot size is just variable and independent of abundance, this can get kind of messy. There are some ways of dealing with it statistically, but it's typically ignored, or at least not specifically addressed in most marine litter publications. Yeah. And high variability combined with sample size can really impact accuracy and precision. So after fitting a negative binomial distribution to the beach litter data that we had, we simulated the effects of sample size on accuracy and precision. So these plots here are the results of 1,000 repetitions or randomly selecting data points from a distribution which mean and shape parameters match that we observed in the field. And what we want to see is that these points are kind of all over the place, and they don't really start flattening out until we're getting up to 1,000 plus beaches. Like, this scale is logarithmic. And so meaning that at least if you want to conduct a standing stock survey, so a snapshot in time of beach litter density in Norway, which is a very heterogeneous, complex coastline, it's really demanding to get an acceptable accuracy. And this is illustrated a little bit if we move away from now the beach litter data and onto the more recently published review paper, that we see some results there highlighting that in terms of from benthic surveys. So this is looking at litter on the seafloor. And this graph here on the left is a density plot which shows the proportion of plots or sampling units that were reported to be polluted in different studies. And not all studies report the same metrics, so the sample size for dive surveys here is really low, so kind of ignore that one. But what you can see is that you have trawl surveys here in the light ones, and we have remote sensing surveys here in the darker, and that these, for seafloor surveys, remote sensing here just implies ROV-based or image-based surveys, and tend to report polluted transects less frequently than do trawl surveys. And surprisingly, it's actually not that uncommon for studies to just not report their plot size in detail, which means that this plot here was like a little bit hard to make, but essentially it's the length versus the width of the transects that were used. And remote sensing surveys tend to be very long and skinny. They can be several kilometers long, but they're done really narrow because the camera needs to be quite close to the seafloor to have adequate resolution for the images, so they can be half a meter, 3 meters wide, but they're quite long and skinny. Trawl surveys are quite variable because there's a lot of different types of trawls out there, but they're also generally long and wider than the ROV-based surveys. Now, because the plots on ROV surveys are so narrow, it's possible that they could miss a patch even if they go through it, depending on the density in that patch, and which can explain the lower detection probability. Of course, it's also a lot less destructive than trawls and can cover a lot of other bottom types. There's a lot of things to consider in here. But in conclusion, there was a lot of variability at multiple scales in both space and time of marine litter, and this variability puts a lot of demand on both research studies and monitoring in terms of replication, and that sort of these patchy distributions really need to be better studied and better considered in a lot of our work. And that was all I had. Thank you.
Thank you, Martha. You made a person who doesn't understand stats understand a bit more stats. So do we have any questions for Martha? I can't see any.
Oh, in the back over there, I think.
Oh, yeah.
You were a little blind, aren't you?
I think I need some new glasses.
I didn't mean that. I meant the lights. Not you specifically. Us up here.
My name is Kim Holmén from the Norwegian Polar Institute. In atmospheric studies, we often replace space with time, and when we have very heterogeneous systems, it sometimes is a useful exercise. Have you studied single spots over time?
I have a bit, yeah. We have. I didn't show any of that here. We have. We are working on. I've done analyses of cleanup data where they clean the same locations over and over, like so part of citizen science data sets. And I've worked on, we have an ongoing mark recapture study in Lofoten where we're really regularly monitoring both what kind of comes and goes and moves around on the beach. And yeah, it is, this presentation focused on space because, time to actually conduct a presentation. But yeah, it is equally variable in time, and sort of temporal autocorrelation is also something that we sort of struggle with coping with a little bit in different analyses and in surveys as well.
I think it really does highlight that complexity that we have and the challenge that we're going to still be fighting for the next few years to understand what's going on with marine litter.
I think so.
Yeah. It's going to be fun, though.
It's going to be fun, yes.
So with that, I can't see any more questions. Thank you again, and I'll invite our next speaker up to the stage. So thanks, Marta. Our next speaker is Emily Cohen, and she's from Syntef Ocean, and I believe she's talking to you today about plastics and the circular economy.
Yes, thank you very much for the warm introduction, Amy, and I'm very excited to talk to you all today. It's a bit of a different sort of presentation in the fact that I work on political and social sciences. I know it's almost lunchtime, but please keep your eyes and ears and mind open for this presentation. And before we begin, I would like to thank the Norwegian Research Council as well as the Environmental Department and my colleagues for making this research come to life. So first and foremost, I want to start by saying that I work both at the global level, but also at the local level in terms of plastic governance. And so starting from the global level, I think all of us know that there are current negotiations ongoing for a global treaty to end plastic pollution. And this will force change, of course, not only from the top, but any sort of global treaty, it will need to be implemented and taken in at the national level, and more so locally. So we need to be prepared for that. I thought it would be interesting to bring to this stage now what was mentioned at the first round of negotiations when it comes to the Arctic. So even though this was the first round where we're kind of understanding the rules of the game, we don't have coherent negotiations going on what's going to be included in the treaty, the Arctic was mentioned twice. Once by Iceland, who mentioned that we already have a lot of important science, specifically when it comes to microplastics and knowledge on creating this global treaty. And the second one was mentioned by the Inuit Circumpolar Council, who as well mentioned science, the fact that we need to use the precautionary principle when moving forward in the treaty negotiations, and also following a human rights-based approach. And I'm going to be following the treaty throughout the next 2 years and see where this flows as well. So back to the study and the project at hand. We ask this research question of what are the systematic barriers in place for preventing a circular economy of plastics in the Arctic. And we use the specific case area and study of Longyearbyen. And this is why we held a workshop in the local community with stakeholders to identify their own best practices forward as well as opportunities. And so before we get into the results of what that workshop was, I thought it would be interesting to bring up what in fact the circular economy is. What do we mean by a life cycle assessment? Because even at the global negotiations, people have different definitions. So I wanted us all to be on the same page here as well. Now, UNEP has their own definition for what the circular economy includes, but I'm going to put it a bit more in layman terms and include a graphic. So by the circular economy, and specifically using a life cycle approach within that, we want to make sure we're looking at materials from the second something is extracted as a raw material, processed, how can we better design products to not have so many materials involved in them, how do we market them, and then eventually moving more downstream to how we recycle products within a circular economy. Now, before we get into the results from the workshop, I wanted to bring in our hypothesis or our presumptions within this project on what the systematic barriers are. And no, it's not this adorable walrus, but he might be a piece of the puzzle. So as we know, the Arctic has limited access to infrastructure and resources, lack of economic incentives, and as well as what we believe to be a limited understanding or public awareness around what the issues are and how stakeholders can contribute to the policymaking process. And so this is why we thought it would be really interesting and vital to integrate stakeholders into our research, and why I think you should implement them into all of your research as well. So let's get into the study at hand. On the local level, we have this lovely picture of Longyearbyen. And it truly does show and seem like this vital ecosystem and town should have zero pollution and zero waste as a top priority. And in fact, we found that it does from a 2020 report from the local government on waste and how they see the future here. We found that in the reports that they believe waste shall be handled in a way which profiles the town as a worthy entrance point to the world's best managed wilderness. And the second goal they had was that the waste management on Longyearbyen should be as good as it is on mainland Norway. So in response to these goals, and with the help of the environmental department here in Norway, we held a workshop on Longyearbyen with around 50 interested locals working in government, industry, tourism, as well as research and even very engaged local citizens. And I also want to point out that citizens are very much stakeholders as well. So what actually goes into our workshops, and how can citizens contribute to this policymaking process? Well, here is a picture, a little blurred out, but that's okay, of how our workshops look. And we use a stepwise qualitative approach in them to facilitate our workshops. And we want to, in any workshop, whatever it may be on, get a full understanding of what the problems are and what the solutions can be from a stakeholder perspective. So we're eliciting information from our stakeholders in a transparent way who live and work in the region, and then also exploring the real world problems and solutions moving forward. So here we have a picture of my lovely colleague who held the workshop. And so we always start out our conceptual mapping workshops with different drivers. And in this case, the drivers are at the top of the screen, seen here. So an example of how a workshop would start is my colleague would point to one of the drivers and say, okay, cost. What do you believe are the financial barriers or opportunities when it comes to implementing a circular economy in the high north here on Longyearbyen? And more often than not, we have lots of hands go up, and after about 2 hours of conversation, making sure we address all the drivers and all of the stakeholders' viewpoints are heard, we have a map where we can see the main points of convergence from our workshop. So now what? What do we do after? Well, after our workshop, we transcribe the audio to make sure that we fully captured the stakeholders' perceptions in the map, and then sometimes we fill in the gaps with semi-structured interviews. And in this case, we held one interview with a stakeholder working on the local government who believed that a major barrier towards implementing a circular economy, having citizens engaged, is that, as many of us know, on Longyearbyen, it's an ever-changing population. Some people could be there for a few weeks, a few months, a few years, but no one ever lives their whole life on Longyearbyen. So that was one of the issues that came up. Another one was with a local industry stakeholder on mainland Norway, and they used to take some of the waste from Longyearbyen, they would clean it, sort it, reuse it, and turn it into new products. However, that as well became too expensive, and they weren't getting good quality materials. So it's important that we have workshops, but we also follow it up with interviews as well. But now, in moving forward to a future where we see this international agreement to end plastic pollution on the horizon, we must ask ourselves, how will this be implemented nationally and more so locally? And so our stakeholders decided on the following points as the most pressing barriers as well as opportunities towards implementing a circular economy on Longyearbyen. The first one being is that there's need for regulatory change when it comes to waste sorting. Currently, Longyearbyen does not have the same regulations that mainland Norway has. A point that the stakeholders brought up in the workshop is that they wish they had deposit return schemes where you can return your PET bottles in the grocery store, and they thought that no matter if it's more expensive to do this on Longyearbyen, it's important to have the opportunity here. The second point was that the stakeholders were really interested in new pilot projects and having a map of all financial resources available for them for these projects. So there was many ideas floating around of how we don't have to export our plastic waste on Longyearbyen. We can reuse them and make them new products on the island. And the last point, I'm almost done, Amy, is that we need spaces to facilitate knowledge and experience sharing. As I mentioned before, there's a lot of population change on Svalbard. And so the stakeholders really said we need a one-stop shop for information leaving or coming into the island. And the last point I want to say is we think we might have all of the answers to research or technology that we're working on, but it's vital that we include stakeholders in our solutions. Thank you.
That was such an interesting presentation. I got carried away and didn't look at the time. So, in the interest of time, we really don't have too many time for questions. So
I'll be around for.
Can we do it in the break
Yes,
afterwards? Would that
definitely.
be okay? Thank you again for your presentation. So our next speaker is, I haven't got their names in front of me, Laurie Ormondson. You've come to the front yet, and she's going to be talking about some of the work you've been doing with port facilities. So take it away. Thank you.
Hello, everyone. My name is Laurie Osmondson, and I'll be speaking about port reception facilities in the Arctic as hubs for plastic waste control. By way of personal background, I am an environmental lawyer by trade. In a former life, I trained as a marine scientist, but somewhere along the way, somehow I took a left turn and ended up in law. But I like to think of environmental law as a combination of science, policy, and legal tools for crafting solutions for environmental problems. Now, we know from previous presentations at this conference, yesterday and today, and lots of research, that plastic in the world's oceans stretches to the polar regions and accumulates here in all parts of the Arctic marine environment, from floating sea ice to the seabed floor and even seabed sediments. Plastic debris is a very visible problem in the Arctic, as well as an unseen one. As shipping and other maritime activities increase in a warming Arctic, stopping the onslaught of marine plastic pollution is a global as well as a regional imperative. Now, negotiations are underway for a new global plastics treaty, but reaching a complete and fully enforce global agreement will still take several years. What happens in the meantime? One present action that can make a substantial difference in reducing Arctic marine plastic litter is increasing the coverage and capacities of port reception facilities, or PRFs. And a recent UN Joint Group of Experts report issued in late 2021 called PRFs the single most effective solution for preventing waste discard at sea. So what are PRFs? A general definition is any fixed or floating or mobile facility capable of receiving waste and residues from ships. Fixed PRFs are static collection points at a port or terminal, usually in a central location. Floating PRFs are normally barges that can be towed or are self-propelled and moved to ships for offloading. And mobile PRFs include land vehicles and movable bins that can be shifted to different ship berths to receive the waste from ships for later pickup by third-party operators. Actually, the upper right picture is a mobile PRF here in Tromsø. So why do PRFs matter for plastic waste control? They matter because they are a primary means of intercepting plastic waste from fishing vessels, from cargo ships, from cruise vessels. And while it's well documented that the lion's share of plastics that have ended up in the oceans over the past several decades have come from land vectors, sea-based sources are far from negligible, and in some parts of the world, including here in the Arctic, they are a substantial vector. We have seen in the last decade a significant increase in ship traffic through the Arctic. This rise is, of course, projected to continue with longer ice-free periods in the Arctic waters and ports over the next decade and beyond. According to the World Port Index database, there are about 350 seaports on the coasts of the Arctic Ocean. It's still unclear, however, how many have functional PRFs for plastic waste. And this is an ongoing, has been an ongoing survey initiative of the Arctic Council to address this data gap. Now, in terms of regulation, MARPOL is the principal international legal agreement for controlling ship-based waste. And Annex 5 of MARPOL concerns garbage, which is defined as including all plastics generated during normal operations of ships. Annex 5 prohibits the discharge of plastics into the sea, and Regulation 8-1 of Annex 5 requires parties to provide adequate facilities at ports and terminals for receiving plastic waste. At present, there's about 155 states that have consented to be bound by Annex 5, and that includes all countries within or bordering Arctic waters. So hence, all Arctic and near-Arctic nations are obligated to provide PRFs to receive plastic shipping waste. Despite these provisions, however, surveys have shown a considerable disparity between volumes of plastic waste that should be offloaded from ships to PRFs and amounts that actually are delivered. A principal challenge with establishing effective PRFs has been what baseline standards should apply, and more to the point, what adequate means. There are some IMO, International Maritime Organization, guidelines that suggest adequate PRFs ought to be ones that vessels actually use and that fully meet the needs of ships using those PRFs, and that don't present disincentives to use, and that contribute to improving the marine environment. Now, those are all helpful suggestions, but what does it actually mean in practice? States have struggled with that, and hence, as a result, implementation has been inconsistent. Now, the European Union issued a new directive on PRFs in 2019, which came into effect in 2021, and it supplies much-needed gap filling on some specifics of creating more effective PRFs. But EU law doesn't apply, of course, to the entire Arctic. So what could be a way forward for the Arctic? In May of 2021, the Arctic Council issued a regional action plan on marine litter. And the action plan sets out a range of strategic activities and measures targeting sources of marine litter, impacts, data collection, monitoring, and mitigation measures. It specifically highlights establishing better and more PRFs in the Arctic to combat marine plastic pollution. And the actions outlined in the plan are to be taken by the Arctic Council in collaboration with governments, indigenous peoples, local communities, and other stakeholders like private industry. Also, we have new amendments to MARPOL, including Annex 5 and associated IMO guidelines for PRFs. These were just formally adopted by the IMO last month. These new MARPOL amendments allow Arctic states for the first time to develop and implement regional arrangements for PRFs, which take into account the unique environmental circumstances and logistical challenges of the region. And the next step with that is joint development of a regional reception facilities plan. And the plan will identify regional ship waste reception centers, and these can include larger, more developed PRFs in near Arctic locations that could serve as mega hubs. So the plan and the regional approach allow Arctic stakeholders to include Arctic-specific parameters for adequacy in terms of PRFs and plastic waste collection from ships. Some metrics of adequacy could include mandatory port waste management plans, sorting of plastics by type, compulsory delivery of plastic waste to PRFs, matching the type of PRFs to the port conditions, transparent indirect fees based on ship class and size, and sustainable, measurable port waste processing standards. Going forward, PRFs can play an important role in facilitating deployment of a circular economy for plastics. As the previous presentation outlined, is an important consideration here in the Arctic. And this could actually complement a new global plastics treaty once it comes online, so that PRFs could serve as hubs in a circular economy and also contribute to increasing economic opportunities for Arctic Arctic communities. And I'll end there. I'm happy to answer any questions. Also, other information on this topic. I have an article just out this month's issue of Marine Policy, and it's also available open access online. Thank you very much.
Thank you, Laurie. That was fantastic. If we can leave the questions to the end, because we've got to move on to the next speaker now. We've got a very tight schedule.
I'll be around.
Yeah, if you just float around at the end, that would be fantastic. Thank you. So our next speaker is joining us online. So if you just bear with us a moment while we sort everything out. It's pre-recorded, sorry. So then I can introduce our next speaker, and she is Kerstin Scherabel, and she's talking about some of the work she's been doing on biodegradability within fishing line and fishing industry.
Hello, my name is Kerstin Scherabel. I am a PhD student at the University of Tromsø, and my PhD project is related to the Center of Research-Based Innovation DSO, which deals with reduction of plastic pollution associated with fisheries and aquaculture. My presentation here is about experiments we conducted regarding potential use of biodegradable plastic materials to reduce the plastic pollution caused by longline fishery. Longlining is a widely used fishing method in different fisheries targeting various fish species. All longlines consist of a mainline, shown here, and hooks that are attached to the mainline by thinner lines called snood lines, which are attached at, as you see, a certain interval here on the mainline. And fish are then attracted to this fishing gear by use of bait on these hooks. In the commercial longline fishery, longlines or sections of them are often lost at sea during fishing because of, for example, being deployed on rough grounds. However, which is maybe more important, during each longline deployment in a fishery, a fraction of these snood lines are being lost at sea. This is because they can get snagged at the seafloor, or when the fish break this snood line during fishing and escape with hook and part of the snood. These snoods are made of non-degradable plastic material, such as nylon. Therefore, this lost fraction of snood lines degrade very slowly in the marine environment. And furthermore, even after a long time at sea, the material doesn't disappear at all, but is being broken down into smaller plastic particles, which increases both macro and microplastic pollution and releases the toxic substances into the marine environment. In general, plastic litter is considered as a major pollution source, and fisheries is being one of the largest sources of the marine plastic pollution. In Norway, demersal longlines are used to target such fish species as cod, haddock, and Greenland halibut. The coastal longline fishery is based on day trips, deploying several mainlines with snoods, such as here in the picture. The number of deployed hooks and snoods range between 10 to 30,000 for each deployment, but for the deep sea fleet this number is higher and can be up to 60,000 snoods per day. As mentioned, a fraction of those then is lost per each deployment. However, the extent of such loss for a longline deployment has not yet been scientifically quantified. To address these challenges related to the plastic pollution, use of biodegradable plastics in different fishing gears are being tested by replacing non-biodegradable materials such as nylon. Compared to, for example, nylon, the biodegradable plastic materials, such as, for example, PBSAT, has the properties for being fully degraded after a specific time at seawater. Therefore, replacing non-degradable materials in longline snood lines could be a potential approach for reducing plastic pollution in longline fishery. However, for this material to be used in snood lines commercially, some characteristics of this new material have to be explored, which is the aim of this study. First, the extent of the snood loss on the mainland during each deployment with biodegradable material should not be higher than that of nylon. This is because higher snood loss would mean more work involved to attach these new snood lines and hooks, which then would result in higher cost and loss in catch efficiency during fishery. Second, the fishing efficiency has to be comparable between gears with biodegradable and nylon snood lines. For example, in earlier experiments using biodegradable materials in different fishing gears such as gill nets showed a significantly reduced efficiency at capturing fish. This would not be acceptable commercially and therefore is important to investigate. Therefore, we conducted the fishing experiments using longlines with biodegradable and nylon snood lines in commercial coastal longline fishery in November 2021. The fishery took place in northern Norway, in Vardø, and was targeting haddock and cod by using baited longlines. During the experiment, we estimated the risk of snood line loss for both normally used nylon material and the new biodegradable ones, and we also compared the catch efficiency for snoods made of biodegradable and nylon material. Furthermore, we compared the biodegradable material with two thicknesses. This is because the biodegradable material has lower breaking strength compared to nylon, and this could potentially show a reduced catch efficiency and increased snood loss because of breaking of those biodegradable snoods. An increased material thickness or the twine diameter of those monofilament snood lines may be therefore needed to compensate for this difference in breaking strength. Therefore, our trials consisted of two experiments. Here, one testing the same twine thicknesses for the nylon snoods, and the second one testing increased diameter of biodegradable material. In each series, the longlines consisted of 6 mainlines, which had 415 snoods each. During the fishery, we observed the fish for each snood line material type. We noted down the species, number of individuals, and length of each fish captured by each snood line type to estimate the catch efficiency of the gears. And then, after each deployment, when the longlines were rebaited and the lost snoods and hooks replaced, we also recorded these numbers to estimate the snood loss. The results of our study is published in Marine Pollution Bulletin. We estimated that the snood loss in this coastal longline fishery when using the standard gear with nylon snood lines is 4.66% during each longline deployment. Well, that would mean that if a vessel used those 10 to 30,000 snoods per deployment as normal in this fishery, then each vessel during each deployment can lose somewhere between 466 to 1,380 nylon snoods, which then contributes to the amount of the lost material in marine environment. When we tested this for biodegradable material, we did not see any differences in the snood loss rate. And furthermore, when we were comparing the catch efficiency for both cod and haddock, we didn't see any significant differences between nylon and biodegradable material for both twine thicknesses the same and the increased one. This means that fishing gear with biodegradable instead of nylon snoods would capture the fish similarly. Therefore, these initial results in this study show that there is a potential to use the biodegradable materials to reduce the plastic pollution in longline fisheries. Specifically, we found no significant differences in either snoot loss rate or the fishing efficiency of longlines, which is a promising result. However, this would further need to be tested in different fisheries and also estimated over longer period of using the material. First of all, I would like to thank you for listening. Unfortunately, I am not able to participate in person, but all the possible questions or comments are very welcome to this email which is shown on this slide. So thanks again, and I hope you have a really nice conference.
Okay, as we're unable to send our questions, if you do have them, just pop them in an email and they can be followed up afterwards. So we're coming to the last speaker of today. We'll take some questions at the end of the speaker as well, and then we're all ready for lunch afterwards. So before I introduce the next speaker, I'll just say thank you for everybody for coming along and taking part in the session. It's been great to have all of the speakers today and on the last day as well. So then I'll invite our last speaker to the stage. This is Anita Parlow, and she's going to be talking on plastic waste from shipping fuels in the marine environment.
Appreciate, and I must say, hip hip hooray that you're all still here. It's kind of incredible. So first, thank you very much. I'd like to say great thanks and appreciation to Arctic Frontiers, Jenny Turton, Anu Fredriksson for inspiring such solid thinking and work on high North Arctic matters. The slides will be backdrop to my remarks, which I will read, forgive me, as to provide context in which the plastic is moving forward. My background is I just recently co-authored a book published a few months ago on climate conflict and food security, mainly subsistence, by Springer Press. And I was involved with a Harvard-MIT project on fisheries moving north, and particularly looking at the Iceland-UK disputes as fisheries move northward, seeking colder waters, and what does that mean as a jurisdictional matter. And so following that, I was invited to help develop a program at the Wilson Center in Washington, D.C. think tank on the IMO Polar Code, and the location was the Bering Sea between Russia, Canada, and the US. It was quite remarkable. And I am a member of the Bar of the United States Supreme Court. So as has been discussed, so now we're talking about waste, plastic waste to shipping fuel. Has been discussed over the past several days. Plastic pollution counts among the 21st century's greatest environmental challenges, a planetary threat damaging ecosystems, human, animal, and mammal health, and the world's oceans. Scientists worldwide report that more than 11 million metric tons of plastic enter the world's oceans every year, with an expected tripling by 2060, according to UNEP. The accumulation of plastics in the world's oceans, beaches, and riverbanks, reflected in enormous gyres such as the Great Pacific Garbage Patch, account for nearly 40% of the world's oceans. The plastic debris spilled from rivers that empty into the oceans, consumer waste deposited directly in the oceans, and 46% of plastic waste from abandoned fishing gear has been expressed. Nets, lines, and traps, referred to as ghost gear, is all dumped into the seas. Ocean scientists document that a range from the tiniest of fish to whales, birds, mammals, and humans experience some combination of intestinal injuries, starvation, and death. Microplastics, often carrying toxins and disease, are likewise ingested by baleen whales or fish and mammals seeking prey. The U.S. Tucson-based Center for Biological Diversity reports that if this annual dumping of plastics is not significantly diminished, there will be as much plastic as fish by weight in the oceans by 2050. Without forward-looking policies at globally coordinated domestic and international levels, plastics recycling, now only at 9% of the 340 million tons annually, according to OECD, will reach only 14% by 2060. It is axiomatic that it should not be burned. Plastic debris increasingly pervasive in the Arctic, often in areas, as has been mentioned, without any human activity or very little. The sources of plastic debris, both large and microplastics, generally originates in the lower latitudes, carried by ocean currents, winds, and rivers, where it generally breaks down into microplastics. This went faster than I thought, accumulating in both terrestrial and marine systems. The microplastics, also created by fibers, enters the food chain to great detriment, given its often carcinogenic and other toxic qualities. Local sources, fisheries, commercial fishing, also impact Arctic ecosystems, where ingestion of plastics, again by all species, are evidenced in dead or dying mammals, seabirds, fish, and whales. China was once the repository for the West's plastic waste, but stopped in 2018 because the plastic was too contaminated to be repurposed. Now, much plastic waste is diverted to the developing world, reflecting an environmental injustice, given the scant available infrastructure to process or recycle the plastic debris. Over the next few decades, some scientists, governments, and business leaders plan to move more aggressively to seek newer, cleaner, and reliable ways to power the world, including the hundreds of millions without any. Companies like Hurtigruten are seeking to power their boats in a manner both fossil-free and renewable. As a matter of revenue to the carbon energy industry, plastic petrochemical waste is and will be insufficient to replace the scope and scale of oil and gas revenues, even if a never-ending spigot of petrochemicals for plastics continues to exist. But from an environmental vantage, plastic to fuels, fuel production process is viewed by a growing number of scientists, commercial interests, and governments as a significant bridge to a greener economy, a stopgap. Norway's Quantafuel, among the first to transform plastics to fuels, uses a patented process called pyrolysis. Pyrolysis breaks down a variety of plastic types through a catalytic system that heats plastic in an oxygen-free environment, converts materials that contain low-value carbon into high-quality synthetic and low-carbon fuel. Quantafuels combines waste plastic with other wastes using steam and heat for a decomposition at elevated temperatures rather than burning the plastic for waste. The outputs of pyrolysis is 80% liquid fuel, the remaining 13% non-condensable gases, and 7% char reused as briquettes in a process designed to lower energy and carbon footprints while cleaning up the plastics at the same time. The Massachusetts-based Worcester Polytechnic Institute is developing a similar approach to convert fuel from plastic on ships at sea using hydrothermal liquefaction. Here, the plastic is broken down into constituent polymers at temperatures up to 550 degrees centigrade and considerable pressures of 27,500 kPa, the standard for measuring pressure and tensile strength. The scientists believe that sufficient fuel can be produced from ocean's plastic to sustain the conversion process, power the ship with an excess for storage, and external markets. Burning the created fuel would generate carbon emissions, but far less than continued reliance on existing mix of fuels. As has been discussed by several persons in the last several days, a series of international initiatives are underway to establish a framework to create legally binding instruments to address the scale and scope of the plastics challenge. One key element is to require plastic chemical manufacturers to provide a full ingredient list. For example, a recent study found that when exposed to sunlight, some plastic types evolve into thousands of new chemical compounds, many toxic and carcinogenic. As for pyrolysis and plastics to fuel oil, the growing number of initiatives offer a logical and solid interregnum until science, government, industry, and individuals produce a better option to fuel the global economy. Thank you.
Thank you. Is there any questions? I'm going to look carefully. Oh, there is actually a question for you at the front here.
Oh, sure.
Hi, and the technology of pyrolysis is also quite controversial, being a very expensive way of producing fuel and also diverting from the real solution, which is actually to reduce plastic production and not having the plastic ending up in nature.
a good point on both counts. One is in terms of the creating a spigot for unending, never-ending plastic. One, the oil and gas industry, it's not profitable enough for them, so it's not going to happen. But two, that the idea is not to create a need for plastic, but to, as an interregnum moment, so a period of time, a decade or so, to clean it up, turn it into fuel, and then move to develop greener capabilities. To your other point about the expense, this is true, and there are two approaches that are being that are underway more or less. One is the pyrolysis process for profit, and the other is a not-for-profit approach. But costs are costs, and if that's what it takes to get plastic out of the ocean and stop killing whales and fish and birds and mammals and little babies, so be it, if I may.
Okay, thank you.
Yeah.
So what I'm going to do is I'm going to close the session and thank you everybody for attending, for the fantastic presentations and posters and interactions that we've been able to have over the last few days. And I'd like to thank my co-chairs. I can't see anyone. I'm sure they are somewhere in the audience. And for everyone for being here. So let's have some more discussions and continue for the rest of the day. And I hope you enjoy the rest of Arctic Frontiers. Thank you.
Han får seg lunsj, sikkert. Tu. Ja. Hvor kom det fra? Hvor vedkommende som sitter og styrer der?